This is part 2 of the chapter 12 recordings. And now we're looking at cytokinesis and cytokinesis in animal cells. So we're going to compare animal cells and plant cells. In animal cells, cytokinesis occurs by a process known as cleavage. Basically, cleavage just is when the cell splits in half. And it starts with the formation of a cleavage furrow, which is a groove in the cell surface, kind of in the middle. And that cleavage furrow just squeezes and squeezes on the center of the cell until it pinches in half. In plant cells, a cell plate forms in the middle of the original parent cell. And then the cell plate eventually forms into the rest of the cell wall. So you can see in animal cells, cleavage furrow where it's indented, and then it just kind of, there's this ring. It's called a contractile ring, and it's composed of microfilaments. So you know what that is. That's the actin. Okay, this is where actin comes in during mitosis. Actin forms a contractile ring, and it just basically squeezes and squeezes until it pinches the cells, and it pinches the parent cell into two daughter cells. And then the plant cell is going to form a cell plate between in the middle of the parent cell, and eventually there will be a cell wall, and you will have your two daughter cells. And you have a video of cytokinesis in an animal cell. And then, so these are on slides 42 and 43. And then you've got some images of plant cells in the 5 different stages of mitosis. And this is under a light microscope. So you could see this. Most likely you're going to be able to see cells that look like this in your mitosis lab, in your lab kit activity. So during prophase, all you're going to see is that you still have, it looks like the nucleus may be breaking down, but it's still there. You know, all of the chromosomes are going to still be in the area of the, of the nucleus, but the nuclear envelope is breaking down, and it's not chromatin anymore. It is chromosomes. Chromosomes can have formed, have condensed. In prometaphase, you're going to see the spindle fibers attached to the chromosomes. In metaphase, they're all going to line up in the middle of the cell. In anaphase, these sister chromatids will separate, and this is telophase. And I can tell that it's telophase because the line in between is very, very faint and light. So it's not a cell wall yet, it's a cell plate. Now, it's not showing you interphase, but I want to show you interphase. This cell is in interphase. You see how the nucleus is like a solid color here? These cells are in interphase, even though they're divided, by the picture, but they're in interphase. And you see how the nucleus is just like a solid blue. That's because the DNA is in the form called chromatin. It hasn't condensed and coiled up into chromosomes yet. So it does that in prophase. So these cells, again, would be in interphase. Now, we're not going to go through this process, but I do want you to know the name of the process of cell division in the prokaryotes. Remember, the prokaryotes are organisms in the domains bacteria and archaea, and they are cells that they're unicellular, and they do not have a nucleus, but they're going to divide by binary fission, which means to split in two. And one of the real differences is the bacterial cell only has one single chromosome. So what you're looking at is a single circular chromosome. It's just, there's a lot of it, so it's kind of bundled all up, wadded all up, I guess. But it's just going to be a single chromosome that duplicates, and then each daughter cell, still gets a single chromosome. There is a control system for the cell cycle that we'll go over a little bit. The cell cycle control system is based on checkpoints. And what I mean by the control system of the cell cycle is, let's say you've got a cell and it undergoes the S phase of interphase. And so it duplicates its chromosomes, and now it's got all its chromosomes have two sister chromatids, right? Well, let's say that when that happens, when the DNA duplicates, the DNA actually makes a mistake. So there can be mistakes in DNA replication, and they can end up causing mutations, which are changes in the DNA code. So let's say that happens, and the change in the DNA code is bad. It's going to be very bad for the cell, the new cells that form. So once that is detected by the control system, it's going to stop the cell cycle at that point. It's not going to let it continue. So we call these different points checkpoints. And there is a checkpoint, it's called the G1 checkpoint, almost at the end of the G1 phase. There is another checkpoint at the end of G2, and that checkpoint would catch a mistake that might have been made during DNA replication. Then there's one more checkpoint called the M checkpoint, and that's going to happen at the end of mitosis. All right, the cell cycle is controlled by two types of regulatory proteins, okay? And they are called cyclins and cyclin-dependent kinases, or CDKs. So these are proteins that help to regulate the cell cycle. CDKs must be attached to a cyclin to be active. Oops. All right. All right, then there is an MPF, which is a maturation promoting factor. That is what it's called when cyclin and CDK are attached to one another. The cyclin CDK complex is called MPF, maturation promoting factor. That means it's going to promote the next stage of cell division. So the three important checkpoints again are G1, G2, and M. Something else that influences cell division are growth factors. And these will stimulate other cells to divide. So growth factors stimulate other cells to divide. There's one called platelet-derived growth factor, and it's made by platelets, which are blood cell fragments that are found within, your blood vessels. So this particular example, PDGF, or platelet-derived growth factor, is required for the division of fibroblasts. Those are the cells that make fibers in connective tissue. They produce fibers like collagen. All right, then there is this, I don't know what the word is, but this condition called density-dependent inhibition. Density-dependent inhibition. And what that means is that crowded cells will stop dividing. Density, high density means crowded. So cells that are very crowded and they're touching each other, they will stop dividing due to that. And it's called density-dependent inhibition. Now, normal cells have it, but cancer cells do not. And we will talk about cancer because that is when mitosis goes out of control. It's uncontrolled cell division. So cancer cells exhibit neither type of regulation of their division. They don't exhibit density-dependent inhibition nor anchorage dependence, which is a different type of control. Well, I guess we're talking about cancer a little bit now, so I will. So when cancer cells are not detected and they're not eliminated by the immune system and they start to grow, they form tumors, which I'm sure you know. If the tumor is benign, it just means it's a growth, but it's not going to spread. So we don't call this cancer. But if the tumor begins to, pieces of it break off and it spreads to surrounding tissues, we call it a malignant tumor. And the process of that tumor spreading is metastasis. We say that the cancer has metastasized when it has spread. And if you find, if you are told by your doctor that you have a malignant tumor, then that is technically cancer. You have cancer. All right, so now I guess what we're going to do now is move on to the M phase or mitotic phase, which covers mitosis and cytokinesis. Oh, this is great. So we could look at all of these cells and remember how we learned that the cell is in interphase for 90% of the time, 90% of the cell cycle. We'll see all of these cells that I'm pointing to right now are in interphase. So most of the cells on this slide of onion root cells are in interphase. Here's some up at the top. But let's look at some of the other phases. This phase is prophase. This is metaphase. This is very early anaphase. This is late anaphase. Let's see. Telophase. Telophase right here. Telophase right here. You can't see the second daughter cell very well, but that's definitely telophase. That's another prophase. Here's a telophase. This one might be at the end of telophase, but I would just say that's two cells have formed here. Cytokinesis is completed here, but this one is telophase. So one more time, prophase, metaphase here. That's the only one I see. Early anaphase, late anaphase. And what it was, telophase here, telophase down here. And I will say, I know you probably noticed I didn't mention prometaphase, and I can't look at a cell under the microscope and tell whether it's in prophase or prometaphase. I will say in prometaphase, you would be able to see that the spindle has formed, and you'd be able to see it kind of looks like a football shape formed by all of those spindle fibers. So you would be able to see that clearly. You can kind of see it here in metaphase, and you can see it here in the anaphase, the early anaphase. You see it's like all these fibers attached to the chromosomes. Well, once you see those, that is going to be where prophase ends and prometaphase begins.